The topic is amplification of gene of interest using PCR. So first in this chapter we are studying how we can create one organism in that organism there will be a DNA. So from another organism we will take the DNA and make it into this organism and we will get the desired character. That will be studying this chapter. So now before that we studied how to isolate the DNA. Now we isolated the DNA and we took from that organism the gene of interest. That means the desired gene which we have to keep into the host organism to get the desired character. So that is known as gene of interest. So amplification means producing multiple copies of the gene of interest, because first we will isolate the gene of interest. If that gene of interest, if we give into one organism only we can give, no, because from one DNA we will get one gene of interest. That one gene of interest we will give to that desired gene we will get one organism, but we have to produce more organisms. So that what we do is we have to multiply that single gene of interest, that desired gene, to many copies. That we will do by a process known as PCR. PCR means polymerase chain reaction. That PCR involves 3 steps. Using these 3 steps we can make multiple copies of DNA. So that this PCR consists of 3 steps. One is denaturation, second one is annealing, third one is extension. So this denaturation means that we know that the DNA is the double-stranded helix. So the desired gene that we took is a DNA, that is double-stranded helix. To create multiple copy, first we should convert the double-stranded helix into single-strands. So that at 94 to 96 degrees Celsius, when we are heating that desired gene, it will split into like this. It will split into two parts, two single-stranded DNA into split. After splitting it into 2 single-stranded DNA, before that, first for this polymerase chain reaction, there are 3 requirements. What are the 3 requirements? Means first one is we need an enzyme. Enzyme is, this is amplification, that is multiplication. For replication of DNA, we need RNA polymerase enzyme. So that is first requirement. Second one is we need a DNA template, because with one DNA template only we can synthesize the next strand. So we need a DNA template. Third one is primer. Primer means it is a short double-stranded, it is a short single-stranded DNA. It is a primer or of 2 types. One is forward primer, second one is reverse primer. Because primer can do this multiplication, that is amplification in one direction. So that we need 2 types of primers. So this denaturation, it will separate into 2 separate strands. In the second process, that is annealing process. Annealing means attaching the primers. This one primer will be attached in this 3' area. Second primer will be attached in this area. These primers are really strands of DNA, but they cannot stand high temperature. So when the annealing process takes place, the temperature will be reduced, or else the primers will be denatured. That's why they will reduce the temperature. After that, the primers, 2 primers are there. In the third step, these 2 primers, with the help of nitrogen bases, they will make the full strand of DNA. It will start replication and it will create full strand of DNA. So totally 2 separate strands of DNA, we will get to 1 strand of DNA. This is known as amplification of gene of interest. That by the process of extension, before that primers will be added. After that, there is an for replication. For replication of DNA, we need DNA polymerase. That DNA polymerase is chat DNA polymerase. That is from Thermosaccharidus T8. This is taken from an archaebacteria. Archaebacteria can stand higher temperatures. So as this reaction is taking place at higher temperatures, they will use this chat polymerase, because biomolecules will be denatured by heat after 40 degrees Celsius. But this can withstand up to 94 degrees Celsius, so that there is the stacked polymerase. This stacked DNA polymerase will attach to this primers at these two places and it will do the replication. This way and this way. Then by the process of extension, these two strands will be produced. So when from one first we had one strand, one gene of interest. From that we got the two gene of interest. If from these two strands, how many gene of interest we will get? 4 will get. So it is in the ratio 2 power n. Means 2 strands we will get. So if n is equal to 1, means if we take 1 gene of interest, we will get 2 strands. If we take the 2 gene of interest, we will get 4 strands.